Functionally, an acid is a chemical that does what? They're in my car.
Oh, good.
This is in the parking lot, I'm not at home. So an acid is a molecule, a chemical that, as you can see by the solutions that are in the beaker, what's it going to do with hydrogen ions? I'll give you a clue. Okay, it's going to release or donate hydrogen ions. Okay, and then a base. So when we talk about a solution being basic or having an alkaline pH, those terms are interchangeable. So a base then is going to do the opposite. It's going to accept a hydrogen ion. Okay. A strong base, as hydrochloric acid is, you can see it on the screen, is not going to be able to not give off the hydrogen. Okay. It is a very low affinity between the hydrogen and the hydrochloric ion. And the polar nature of water, remember with its positive charges on the hydrogen side and the negative charges on the oxygen, has enough electrical effect to separate those positive and negative charges with that hydrogen chloride ions to separate that bond, that ionic bond. And so the hydrochloric acid easily dissociates in solution and gives out a hydrogen ion. So remember, we determine acid by the number of 3 hydrogen ions, not the ions that are bound to some other component of the molecule. So carbonic acid, I said, was a weaker acid because when I spent about 15 minutes trying to find a site last night on the internet that would tell me the relative numbers, and I got a whole bunch of chemical equations and pKs, and I didn't want to take the time to sit down and figure out actual numbers. So I'm still searching for that. But anyway, carbonic acid has a higher affinity for the hydrogen, and so it's less easily dissociated by the attraction of the positive and negative charges in water. Yes?
Do you remember what the pH was for carbonic acid?
That's what I was going to do next, was look it up. Actually, I thought of that on my way to work this morning, and then I did something else, but that's, yeah. But that would give you a, look it up on your smartphone. So, when we, why are our normal range of pH in the body 7.35 to 7.45. All right, so a slightly alkaline pH. Why is pH regulation so important? We've talked about a few things so far this semester, mostly in the last month, that were affected some last semester, that are affected by additional hydrogen ions, or more so than most cases.
And then?
Okay, proteins. So protein structure is going to be affected by the charge. And so we have enzymes that would not be able to recognize their substrate. We have the binding of hemoglobin and oxygen that's affected. Receptor on the surface of a cell, recognizing its other markers on other cells or antibody components. What about with nerve function? Anybody remember what happened to the resting membrane potential if we surround a cell with hydrogen ions? Does it depolarize or hyperpolarize? I know. In fact, I thought you were done with the nervous system, and I keep bringing it up. So we normally have positive ions inside the cell, but just that are potassium, but just under the membrane we have a lower number of positive ions proportional to the outer number. And so typically when we open up, the channels, we tend to add sodium ions. All right. However, if we add we have leaking potassium ions going across the membrane, going against the electrical gradient, positive to more positive. And so the potassium ions do not leave as much, and so yes, our resting membrane moves higher, and the cell becomes more irritable, becomes easier for that cell to depolarize. All right, so that's the effect of having additional hydrogen ions. And the opposite effect, not having enough positive ions around the cell would lower the resting membrane potential, and we lose more potassium and make it more difficult. So the body has an interest, to use the term, in maintaining a normal pH, right? And the structure that we use to do so, when we want to balance pH by adding hydrogen ions when there's not enough, and by removing hydrogen ions when there are too many, is a term then known as a buffer. So what does a buffer need to be able to do to be able to adjust pH? Because that's the role of a buffer. It adjusts the pH or hydrogen ion concentration by doing what? Does it only buffer acids, or does it only buffer bases? It has to work in both types of solutions. So in an acidic solution, a buffer is going to give off a hydrogen or accept a hydrogen? Accept. It's going to act like a base. And in a basic solution, it's going to act like an acid, and it's going to donate a hydrogen ion. So typically buffers are weak acids or weak bases. Why would a strong acid not work?
They don't want to share their hydrogen.
They would always give it up. Okay, you can see in the hydrochloric acid, it's not going to accept it back again. It has such a low association with that hydrogen ion that there's no way it's going to take it back up again. You can have a lot of hydrogen ions in solution, and the chloride would never take back the hydrogen. It's always going to just dissociate. All right, so that's why it needs to be a weak acid or base to be able to function as a buffer. So we are going to look at 3 different mechanisms by which the body buffers. We have chemical buffers. We have the respiratory system. And we have the urinary system. And there's a bit of overlap in these 3. So here's a distribution of chemical buffers. So on the right side are buffers that we find inside cells. And on the left side, buffers that we find in extracellular fluid, such as interstitial fluid or blood. Okay. Phosphate buffer system is a very important buffer system inside cells. And as you'll see, it's also very important in the urinary system. So we have phosphate buffers. Cell and urinary filtrate. And protein buffers. And see on the board, a strong example of a protein buffer is hemoglobin's role in buffering the hydrogen ions that are released when we synthesize carbonic acid inside the red blood cell using carbonic anhydrase. But we have proteins also in our plasma, made by the liver, and a variety of amino acids, and then we have carbonic acid. So the strongest ones for extracellular fluid would be proteins and the carbonic acid component. Now, carbonic acid, a lot of students confuse with the respiratory system's role in buffering it. That's where we see an overlay, okay? can also function with other aspects. We don't need it just with the respiratory system. We have bicarbonates formed in other components. Urinary, not digestive system. The pancreas releases bicarbonate ions. Okay, but it does have an overlay with the respiratory system. So proteins and carbonic acid. So let's look at some of these chemicals first, and then we'll come back and review the respiratory system, and then focus on urinary system, which I've been promising for a couple of days we would talk about. So here is, in the center of the screen, an amino acid. Okay? Amino acids, I'm going to go to the middle board here. Amino acids have two ends. They are an acid, so they have a amino acid component, and this is our acid molecules when we talk about fatty acids. Fatty acid tails have that COOH, so that's where the acid component comes from. And the R means, depending on the amino acid, there can be other components to it, or it can be attached to another amino acid in a protein. And then we have the amine end, okay, for the amino acid. The amine has to do with the nitrogen. Okay, now, in a neutral pH, all right, the amino acid looks like this. Now, depending on the protein, all right, they will have a variety of different forms of this at different pHs. So that's variable with each amino acid on it. So it has its own particular pH at which it shows distribution of the negative and positive charges. Just looking at the function of a protein. Now let's say that we are in a solution that has lots and lots of hydrogen ions. Okay, our pH is acidic. In this type of a condition, would this hydrogen ion come off? No. Now, this is not regulated by hormones or enzymes. It has to do with concentration drive, okay? Technically, yes, it would come off. But we have so many hydrogen ions in solution that it would be immediately replaced by another one, okay? So in essence, you wouldn't know that it had come off. So in an acidic solution, this hydrogen ion is going to be present, and then the nitrogen is going to accept a hydrogen ion. So this would be the acidic view. So if you looked at a molecule that looked like that, you would know that the surrounding solution was acidic because it's driven the hydrogen to bind to that nitrogen group. Now, in a basic solution, we would have fewer hydrogens. We obviously wouldn't have one bound there because there's so few hydrogens that it's not going to be a stable interaction. And so in that solution, type of a solution, this hydrogen comes off as well. So we have a substance here, a compound, that either accepts the hydrogen or donates a hydrogen, depending on the number of surrounding hydrogens in solution.
Didn't you just donate a hydrogen to get the negative?
Well, this is not that nitrogen, the hydrogen that was on the nitrogen is not normally there. So this is our neutral view, so then if we become more basic, this hydrogen comes off.
What about the one down
If it accepts
there?
the hydrogen,
I think
then isn't that the definition of the base? The nitrogen accepted the
No,
hydrogen.
what I'm saying is in a basic solution,
The amino acid donates it.
the
Amino acid.
acid side of this is going to donate a hydrogen. So in a buffer, okay, so I'm not saying that this is a base. I'm saying that this is a buffer.
Okay.
And in a basic solution where we don't have enough hydrogen ions, it's going to release a hydrogen to add to that solution and lower the pH by raising the number of hydrogen ions. Does that make sense? Okay. So what was here comes off and adds its hydrogen charge, positive charge, to the overall solution. I'm going back and just changing the slides.
Oh. Sorry, everybody.
So here's the carbonic acid chemical, and you guys already know this. So in this case, the carbonic acid is the weak acid, whereas here it was the amino acid that was the weak acid. So that was our protein, amino acid of our protein, and you're already familiar with a carbonic acid component, but I'll write that on the board as well. So now if we put, if you were to see a solution that looked like a high number of molecules that looked like this, would you assume that the solution was acidic or basic? Basic. Does that make sense? It seems backwards because this is a bicarbonate ion that accepts hydrogens, okay? But the reason it's missing its hydrogen is for the same reason this one is missing its hydrogen. It's in a solution of low numbers of hydrogens, and so it will have given off its hydrogen. I'm not writing this in a chemical formula here. But it would have given off that hydrogen to add it to a solution that didn't have enough. Okay? And then again, if it was in an acidic solution, so if we change this solution to acidic, we have too many hydrogen ions. We would add a hydrogen back to here. We would add this hydrogen back, and then we would change that to that by adding a hydrogen back to that, and that would help to remove some of the hydrogens that are in solution. Okay? So phosphate is also a buffer, and as I indicated, it's a very strong buffer in cells, but here we're seeing it functioning as a buffer in solution, in the urinary solution. Okay. So as if we were to see that phosphate as H2PO4, would you assume that we were in an acidic solution or an alkaline solution? Acidic. Okay? Because then if it's giving off a hydrogen, it looks like that. Okay? So you can figure out knowing what so this is what we mean by a buffer, being able to either accept or give up a hydrogen. If it always gives up, or it always accepts, it's a strong acid or base, and it doesn't work as a buffer. Okay? And we'll come back to this image in a moment, because we're going to be buffering hydrogen ions in the filtrate. Okay, so that takes care of our chemical buffers, and like I said, we see an overlap. And now let's look at the respiratory system. Okay, I'm going to leave that up there for when I come back to the urinary system. The respiratory system is not a chemical buffer. It uses carbonic acid, but it is a behavioral change. So what is it? We change our respiratory rate. So when we use the respiratory system to buffer, rather than talking about adding or removing hydrogen ions from a chemical, what we do is we increase respiratory rate and depth, or we decrease respiratory rate and depth. And that's how we use the respiratory system to decrease or increase the number of hydrogen ions that we have. So let's say that one of the causes of ketoacidosis for diabetics is they don't have glucose available in their cells. used for fuel. So they start breaking down lipids, and they take fatty acids, which are long chains of carbons. And they break them down into 2 carbon groups. If you've had micro, you've probably talked about beta oxidation. If you haven't, we'll do it in a couple weeks when our next or our last lecture. And so one of the byproducts of this is an increase in the fatty acids. And so that condition is known as ketoacidosis. What would be the change in respiratory rate if your pH dropped, so you have too many hydrogen ions and a low pH? What would be the change in your respiratory rate? Would it increase or decrease? It's going to increase, all right? So your response would be an increase in respiratory rate and death. Blow off CO2. And because of the buffering, because of the aspect of carbonic acid that we talked about, a decrease in CO2 is going to equal a decrease in carbonic acid formation, and a decrease in carbonic acid formation is going to equal a decrease in hydrogen ions. So we don't actually get rid of hydrogen ions by increasing our respiratory rate, but we get rid of the CO2 that can downstream increase the number of hydrogen ions. Any question?
I'm following your chart.
Okay. All right? So that's kind of the behavioral change that helps us to adjust the pH. If we talk about, let's say someone's hyperventilating, it's one of the very difficult to go into respiratory alkalosis.
It's increase of CO2.
So with hyperventilation,
Increase of CO2. Increase of hydrogen bicarbonate.
we are increasing the pH. Well, I don't want to do respiratory yet. Sorry, guys, let me change that. Let's say that excessive vomiting. Loss of hydrochloric acid from the stomach. Which you might think is a good thing. But the body synthesizes bicarbonate ions in the pancreas, and we absorb them across the intestinal wall. So now we've absorbed all those bicarbonate ions, but we don't have enough hydrogen ions to balance them. So we have too many bicarbonate ions. So our pH is too high because we, rather than having too many hydrogen ions, we really have too many bicarbonate ions. So the respiratory response, we need additional hydrogen ions to neutralize our bicarbonates. So the respiratory response is going to be to decrease our respiratory rate, have a more shallow breathing, and that will cause us to retain carbon dioxide, and an increase in carbon dioxide will equal an increase in carbonic acid, which will equal an increase in hydrogen ions. And those higher hydrogen ions will bind to our bicarbs and neutralize them. Get them out of solution. So that's a respiratory change that helps to adjust pH. So that's how the respiratory system helps the body modify pH. Now, both of these causes of change in pH are considered to be metabolic changes, metabolic causes. Now, obviously, we can never stop breathing, okay? So we cannot completely adjust our pH. Sometimes times by slowing down our respiration, because our breathing is required to get oxygen. So you can't just take 1 breath a minute and retain so much CO2, okay, because then that's going to not provide you enough oxygen, but it is a help. Other causes, when I started to write on the board, other causes of change in pH are respiratory. So can I erase this part of the board now?
Is there a name for the excessive vomiting?
No, you mean emesis is another word for vomiting. Okay.
Thank you.
All right, so may I erase this? And we're going to look up at respiratory causes. Nobody said yes, but nobody said no.
E-N-E-S-I-S.
So instead of metabolic causes, we'll look at respiratory causes. So pneumonia. Pneumonia is going to cause an increase in carbon dioxide, which is going to cause an increase in carbonic acid, which is going to cause an increase in hydrogen ions, and a drop in pH. So if we follow the logic and activities that I just erased from the board, then to buffer this with the respiratory system, we would increase our respirations, okay? Which your body probably does, but never works. Because the cause is a respiratory problem, we can't fix it with a respiratory change, okay? So buffering, respiratory system buffering is ineffective when the cause is a respiratory problem. That's our next one. So we talked about the respiratory change. Now the third system is urinary. Unfortunately, urinary is effective in both causes. So urinary system can be effective in adjusting pH for whether it's a respiratory cause or whether it's a metabolic cause. So most of the cells in the kidney that are involved in adjusting pH are active during acidosis. So they are going to be moving hydrogen ions. Are they going to secrete hydrogen or reabsorb hydrogen if they are involved in adjusting a pH under acidotic conditions? What's our definition of secretion and reabsorption? Secretion is adding to which fluid? The filtrate. So if we have respiratory acidosis, are we going to secrete hydrogen ions or are we going to reabsorb hydrogen ions? We're going to secrete. Okay. So the diagram that I'm going to draw on the board, I'm going to go through it twice. I'll go through it with the pumps and channels that are effective. That makes it a bit messy, especially when I did it over there in the black wouldn't erase. It's like seeing double exposed pictures. And then I'll erase the cell and just look at the base. Well, maybe I should do that first. I'll do that first. I'll just go through the basic movement, and then we'll see what ion channels it goes through. Okay, the channels are shown up there, the antiports and pumps and so on. So we have in the kidney, and this takes place in the proximal convoluted tubule primarily, the ascending limb of the nephron loop, and the distal tubule. Okay. It says up there collecting ducts. There are some cells also in the collecting ducts. And this is the primary direction. At the end of this discussion, there are a few cells in the collecting ducts that will be active during alkalosis. All right, they will absorb, they will do reabsorption of hydrogen rather than secretion of hydrogen. But the primary transport that occurs is moving hydrogen into the filtrate. So be very basic at first. We have a tubular cell. And we're looking at the proximal convoluted tubular cell here, just to name it. All right. Over here will be peritubular blood. And over here will be filtering. So I'm leaving out all the fancy microvilli and all of those. So our problem is acidosis. We have too many hydrogen ions in the blood. Right? So we are in acidosis. And for the urinary system, it doesn't matter if the acidosis is due to pneumonia and not being able to excrete carbon dioxide or transfuse, diffuse carbon dioxide, or if the problem is diarrhea and getting rid of the bicarb ions or ketoacidosis. Just too many hydrogen ions. Because our proximal tubular epithelium is identical to our tight junctions in the blood-brain barrier in the nervous tissue, hydrogen ions will not cross the basolateral membrane. And bicarbonate ions will cross the apical membrane. So how did we get around this problem when we were looking at trying to activate central chemoreceptors? CO2. Okay, so we do the same thing in the kidney. We take hydrogen ions plus bicarbonate ions, form carbonic acid. You guys know this by heart now, right? That forms carbon dioxide and water. That's what's happening in the purple section up there on the screen.
Oh.
CO2 is moving, well actually it's not shown, it would be happening in a capillary, but what's happening in the purple section now is the carbon dioxide is going to diffuse out of the blood into our tubular cell. And within the tubular cell, carbon dioxide is going to react with water. And here we have carbonic anhydrase. So it's going to drive
It just reverses.
the reaction. The reverse occurs. We make bicarbonate ion and hydrogen ion. And the hydrogen ion ends up in the filtrate. Bicarbonate ion goes back to the blood and returns the carbon dioxide and the water ion back to the blood where it came from. So we've taken a hydrogen ion, moved it into the filtrate, and returned the rest of that back to the peritubular capillary. So this is, I'm sorry guys, my day's been too long. Well, I'll leave it. This is what happens, but I was going to put in a transport switch, and I'll leave it there until later. All right, so what we've done is secreted this hydrogen ion from blood to filtrate, right? That's what we talked about as secretion. It's moving from filtrate to blood. So we have fewer hydrogen ions in the blood, more hydrogen ions in the filtrate. So the blood pH should rise, and the filtrate pH should fall. Okay? Again, I'm doing this without the channels. I'll do it again. It'll look more like what you see on the screen. All right, that's just part from the filtrate and send them back to the blood and use them in the blood to buffer even more hydrogen ions. So we can use two different things to lower the pH of the blood. We can get rid of hydrogen ions by secreting them into the filtrate, plus we can buffer additional hydrogen ions in the blood by taking bicarbonate out of the filtrate. And we just do the process in reverse. So this is secreted hydrogen ions. All right, now let's look at bicarbonate ions, which are filtered. So this came through the filtration membrane, but it can't cross the apical membrane. All right. So we just do the same. We add hydrogen ions, form carbonic acid, that dissociates into water, and carbon dioxide. Carbon dioxide diffuses into the cell, follows the same pathway, using the same carbonic anhydrase, and now the carbon dioxide moves out to the cell, and the hydrogen ion moves back to the filtrate. So we overcome those basolateral and apical membrane blocks by taking bicarbonate ion from the filtrate and reabsorbing it back into the blood. So now this is reabsorption. Taking hydrogen ions from the blood, converting them to carbonic acid, ending up with hydrogen in the filtrate. Taking bicarbonate ions in the filtrate, converting it to carbonic acid, and ending up with bicarbonate ions in the blood.
Looks like it all cancels each other out and just like this again.
Okay, what we end up with, yes, but remember this CO3 came from CO2 that was already here. Okay? So what we've done is actually added new bicarb and new hydrogen. So yeah, it looks like it's the same chemical, but we took a hydrogen ion. The reason it looks the same is because we go through the same sequence. I could have written that as a separate. So if I write that as a separate pathway, now our CO2 plus water forms carbonic acid. We send the hydrogen ion back here and put in a new bicarb. Okay? And we would use carbonic anhydrase to do that. So now we have double the effect. We've gained a new hydrogen and lost the bicarb. So we lower the pH of the filtrate by two mechanisms. Losing bicarb, but gaining hydrogen. Most importantly, which is what our purpose was, we've lowered the pH, we've elevated the pH of the blood by losing hydrogen, plus we gain another bicarbonate that we can buffer another hydrogen. Okay? So I'm going to leave that diagram there, and I'm going to redraw the process on this board, but putting in some of the pumps, okay, some of the transport molecules.